Hi, welcome back. General Ecology. And we're now entering into, I think, some of the more interesting parts of ecology, and that is species interactions. To me, that's what really gets me up in the daytime, getting up and thinking about how species interact. They don't behave in isolation in petri dishes or on computer models, although we're going to use modeling some more. They're really doing things to, for, and with each other. And I am fascinated by that. So chapter 14 is about predation and herbivory and the impacts of those on population dynamics. So here's a nice picture of a lynx, and it's catching, in theory. I can't imagine that poor little hare is going to get away, snowshoe hare. And classic study of predation cycling with lynx and snowshoe hare. I'll show you some of those data later, but it's really how a lot of this got started from the Hudson Bay Fur Trading Company and their records of the furs that they were collecting. And they were collecting them in kind of like those gyre falcons in cycles, because that's what the animals were doing. They were having boom-bust cycles. Largely a function of the animals themselves and the environment they lived in. This is not a new thing, and I throw this slide here from chapter 13. You know this pattern, and I want you just to see it. So these are, there's a study up on Isle Royale looking at moose and wolves, and it's been going on for many years. It's an island, and fantastic, highly dissected system. But this figure shows you a really nice example where the blue line is the number of wolves and the green line is the number of moose. Okay? And what you hopefully are seeing is that whenever the moose are peaking, the wolves are not. Okay? So here's the moose. It's not a huge peak, but look where the wolves are, the blue line. Here are the here are the wolves here, and look where the moose are. Here are the wolves here, and look where the moose are. And then they use up their resources, and then the moose can rebound. There aren't enough wolves to eat them. The bottom line is that they are offset. You're not going to get both populations at their peak at the same time, typically, because they are impacting each other, right? Resource bases and or predators. So we're going to look at that. And in this chapter, predators and herbivores can limit the abundance of each other. We can switch the words around to predators. Instead of predators and herbivores, we could say consumers and consumed. And they can cause fluctuations in regular cycles. And then predation and herbivory favor evolution of defenses. And that gets kind of fun towards the end, some nice images of how organisms defend themselves against herbivory or predation. So here's a nice case study that I like a lot from Australia. They introduced prickly pear in Australia to help protect their crops. So they planted them and they spread like wildfire. Cactus only occurs in North America. Even those euphorbs that you have from Home Depot that look like a cactus and have little flowers and little leaves occasionally, like the crown of thorns, those are euphorbs. They've converged on the cactus form, or the cactus form's converged on their form, I don't know. But those are not true cactus. Those only occur in North America. This image on the left shows you prickly pear from probably Mexico, maybe Southern California, that have spread and become a nuisance. They didn't know how to get rid of the stuff. It is no fun to walk through, either as cattle, kangaroo, human. You don't want that stuff. So they were trying to figure out how to get rid of it. It propagates super easy, both by underground propagation and seed. And then a third thing, those big fat leaves can just break off and then reestablish a new plant. So really insidious little weed. If you look at those figures, you can see these little placards with numbers, C15, C16 on the left side. Here they are a couple years later, C15, C16 over here. But note the prickly pear is largely gone. Why? Because of that little moth, the cactus moth. So they went back to Mexico at some point and they found out that this cactus moth was controlling the populations in their native habitats, and they brought it over and released it. In this case, happily successful. Not only did it work, but it also, the moth didn't spread to other plants in the area like natives that would become endangered as a result of the introduction. Failed introductions are very common where people release things and then it creates even worse problems than they thought. Classic example I'm sure you're aware of is the mongoose being released to get rid of the rats in Hawaii, but they don't see each other. One is asleep when the other is out hunting. So anyway, this one worked, and in addition to the fact that it didn't spread to other native plants, it basically is able to maintain a population so that when there's an outbreak of the cactus, the moth eventually finds the cactus and knocks out that little population. So it's in a dynamic state now, but it's much better than it used to be. We still messed up, but at least we had some impact on controlling it, the problem. So predators are very much an important part of the system. This figure shows you anole lizards and orb weaver spiders. And these researchers noticed at some point that where they found spiders, there was probably many less lizards on that particular island. And so they did a nice little controlled manipulative experiment. They put 20 individuals of orb weaving spiders onto 5 islands with lizards, and 20 individuals each onto 5 islands without lizards. And that's what the data you're seeing there. You can see that islands without lizards, the spiders on the y-axis, going from 0, that is when they introduced the spiders, or the spiders, out to 5 years. Without lizards, the orb weaver spiders did just fine. Well, they got into sort of potentially a population cycling. Maybe there's some concept of a carrying capacity there, although each island is probably a different size, so I think it's probably overshooting that idea. Note the humongous error bars. That's okay. A lot of students get really flipped out when they do their error bars and they see these kinds of things. They say, oh, we did something wrong. No, there's a tremendous amount of variation in the system, and you've documented it. That's fine. Don't worry. It's really fine. I mean, it is what it is, in other words. But it's really fine if you compare it to the islands with lizards. There's no overlap, right? So there's not even a hint that there's a lack of significance here. Definitely a significant pattern. Great experiment. Manipulative. They were able to follow it out for 5 years, and they saw a beautiful treatment effect. That is, lizards definitely eat orb spiders, right? So 10 times more abundant when there were no lizards. When there were no lizards on the island. Okay. Looking at this a little bit more, here's another case where a little bit of knowledge goes a long way for controlling an invasive species. In this case, this little flowering plant, well, it's a shrub called St. John's wort. You can actually buy supplements of this stuff in the store. I have been in, okay, here's a little crazy story of Horton. I was at a conference in Sweden. This is the Mycorrhizae conference, people that study fungi. And we actually started the first one when I was doing my PhD at Berkeley. And at that meeting, we started a social event where we would bring wine to a party, I think midway through the conference. It's like a 4-day conference. So probably the second or third night. and you brought a wine from your home and shared it with people. And then it was in a bag so people couldn't see it, and it was rated. And it became just a silly, crazy thing because people get really over the top. My wine's the best wine ever, and your country doesn't make any good wine. Why are you bringing... Okay, it gets fun. So I was walking around with my wine, and this old guy named Torgny, who was Swedish, came up with this bottle, and he pulled it out of his bag and showed it to me, and it was maroon, cloudy red. And I know enough about wine to know that that's not what it should be. And he said, oh, this was, yeah, anyway, he said, this, you'll love this. This is really good. I said, does it taste good? He said, yes, it's from Sweden. I'm like, okay, I hadn't thought of you as a wine-producing area. What is it? He said, I'll tell you. It's St. John's wort extracted from my backyard. Now, Torgny, mind you, was about 80 years old. He was at least in his 70s. Real nice man. I had gotten to know him in previous meetings. I said, really? He said, yes. This will help your hangover. And I'm thinking, all the wine that I've been drinking of different people's, of varying quality, I thought this actually might be worthwhile. And being the crazy guy that I am and trusting Torgny, I went ahead and had a sip, a small glass of it. His did not win the award. I forgot whose did. But I didn't have a hangover the next morning. Absolutely confident it was the St. John's wort, not all the water I drank and reduction of wine after a while. But anyway, St. John's wort is actually medicinal. You probably can find it in your supplements if you look carefully. Unfortunately, it's also a terrible pest for fodder, as fodder for cattle. They actually are poisoned by it to some degree. Perhaps humans are too. I'll let you decide. Also called Klamath weed, introduced in the early 1900s out in California. And in the 1950s, biologists figured this out and introduced a leaf-feeding beetle that consumes the weed. And it worked. Look at that figure. So the green is the Klamath weed, right? From 51, it was, boy, there was so much percent cover, 80% cover, just swamping out the pastures. And then the introduction of the beetle comes in and crash. And now it's in some sort of stable cycling. So yes, you can find St. John's wort, but you can also find the beetle out there. And it eliminated 99% of the Klamath weed populations in North America, not just California. So a nice success story. That's also been practiced with purple loosestrife, I believe, and others. You do always have to be careful, though, that the thing that's going to knock down your pest doesn't switch to a native species when it has finished eating the one you want it to. So this is a big problem with invasive species management. Okay. Populations of consumers and consumed populations fluctuate in regular cycles. We kind of started to see that here, right? You have your pest, you have, or I should say, your resource, the Klamath weed, and then you release that beetle that eats that resource up, and it's a delay, but it peaks, and then it tanks, and they kind of follow each other, presumably from then on. And here is the classic version of that. This is predator-prey cycling, in sync, but offset, right? And these are the hare and lynx, snowshoe hare and lynx, from the Hudson Bay Fur Trading Company that went back many, at least 100 years before the 1850s. And so the snowshoe hare is in blue. You get a peak in the population of the snowshoe hare, and that feeds the lynx growth, who then find and eat all the hare and bring that population down, which then starves out the lynx. Now the hare is able to be released, and their population goes up. Now the lynx has more resources because it crashed. Now it's gaining again until it wipes out the hare, and it just keeps going, offset by a couple of years between the hare and the lynx populations. Classic, and really the beginning of understanding these things. that is population cycles and predator-prey. So at some point, a guy named Gauss, who you probably have run into if you have done any kind of math work, and he created a predator-prey cycle in lab, which is pretty cool. A little contrived, as you'll see. So it took another person to come along and tweak that contrivance and actually get it to stabilize and work. So let me walk you through this. So Gauss is using paramecium as his prey. That's the thing on the left. You've seen that in your petri dishes in lab at some point, I'm sure. And then he used didynum, or is that didynum? Not my world, on the right. And that's a predator. So paramecium are the resource, didynum is the predator. In the first experiment, he put both into a beaker with lots of nutrients. The predators ate all the prey, and both went extinct because there were no longer any prey left to eat. These guys go extinct, and these guys were gone already because they'd been eaten. So he says, okay, I'll give the prey refuge. And he put what essentially looks like cotton, but it's glass wool, little strands of glass, super thin. The cotton would get all matted with water, so the glass wool holds its shape. And now the paramecium can find their way into that glass wool and hide out. So again, the predator eats everything he could find, or she can find, or it can find. And then it went extinct because it could no longer find any paramecium. However, the paramecium a few, were hiding, and they rebounded. So they came back. No more predators left in that petri dish, though. So the cycling didn't happen in that case. So the next experiment was say, okay, have the refuge, have the predators and the prey, but now after each round where the predator goes extinct because it's eaten up all the prey, and some prey are left to rebound, now have a couple of predators reintroduced into the petri dish. And he called it immigration, which I think is funny. So in other words, he's tinkering. He says, there are no more predators, the prey is rebounding. Well, I can make this cycle by adding a few predators back in. Not sure if I was his advisor and he was my student that I would find that to be wholly satisfying, but I get it. It makes sense. And this was the first time it was done. And you could see that, by adding predators back in, you get this beautiful population cycling. It's a little bit contrived. So this just shows you the same data in a slightly different, more simplified version. In experiment 1, you have lots of prey. You add the predator, they eat the prey. Prey goes extinct, and eventually the predator goes extinct too because everybody's eaten in that situation. Then if you give some glass wool, the predator will go extinct, but a few prey survive in the glass wool, and then they rebound and become overpopulated, right? Back to dNTT equals Rn. And then if you add a few predators every once in a while towards the end of the prey population increase, predators have gone extinct, but now you can add a few more and they take off and take care of the new prey that have developed after they came out of the glass wool. And now he got this nice cycling, although he's manipulating it in a big way, as I suggested. Which takes us to Huffaker's experiment. And I love this one. It's a little complicated. If you're not already a little bit flummoxed, I make sure you just come back to these things and look at them again. Huffaker had a table in the lab with rubber balls that are darkly colored in this figure, and orange balls, which are actually oranges in this figure. There were a few oranges, lots of rubber balls, and then he probably had Vaseline around here so that his mites wouldn't escape too much. This is actually in the building where my department office was at Berkeley. Huffaker was a professor at UC Berkeley. And he was working with, it was in plant pathology, or in this case entomology probably, insects. And it was a pest, these mites were pests on oranges. in the California and probably Florida as well. And he wanted to see if he could get population cycling with the prey, which was this one, I believe the six-spotted mite, and then the predator over here, which is no name given. The predator could only walk between oranges. So if it got up onto an orange, it had to walk down, get onto the ground, and then walk over to another orange. All right? The prey could put out a little thread of web or silk thread, just like a spider. I mean, they're somewhat related, arachnids. And that was enough to, with a little bit of wind current, carry the mite. Let's say it's over here, puts out its thread, and then the wind carries it over to another location. And so the prey can escape, the predators can follow, but a lot slower. They can only walk. That's the setup of this experiment. And then here are the data in a cartoon form. And again, let me walk you through this. It's pretty cool, but you do have to sort of slow down and observe what's being shown. So the shading up here in these boxes tells you how many prey are in that little sector. So if it's dark red, there's a lot of prey. If it's a little less, more like what color is that? And tan. There's a lot if it's red, less if it's tan, and virtually none if it's white. The black dots tell you a predator is there. Okay, so once a predator is there, then any prey that are there are going to try to escape or get eaten. And then the cycling here. Notice he's got these cycles at roughly 100-day cycles. All right, and so, is that right, 100? Yeah, I think so. 75 maybe. And so in this first phase right here, on the left, there's a lot of prey. And there's a lot of prey kind of all over. But look at this upper left-hand corner. There is a lot of prey, but there's a lot of predators. And in the next 50 days or so, turns out those predators are all alone. The prey have escaped. And they've dispersed out around the rest of the table. A bunch of them were down here already, and the predators were still there, and they got hit once they got there. But again, these then are starting to escape and find their way where there's empty sectors. So look at this one at, I guess this is day 75. The prey is starting to rebound all over. The predator has died off. There are a few down in here, but not many up here. And now you can see in the next phase, the prey really take off. Also down in this lower corner, although the predators have found them. And you could just kind of keep going. And then if you put that into the population cycle curves, you see that's exactly what's going on. So prey is red, they go up, the predators follow. Prey go down, either through escape or being eaten, and eventually the predator mites starve because there's no more prey around in that area. And it keeps cycling like that. And he got this to go on its own, no additional animals added, for over 200 days. And I see that as a pretty profound success in this particular field. I really love that. Simple experiment using mites, and was able to show that the predator can be used to control the populations of the prey, which was a problem for orange growers. And it was a sustainable thing, just like we saw with the Klamath weed and prickly pear at the beginning of this. So I'm going to bring it to a close here for now. We're going to take up this chapter and this lecture next time we meet. I believe what I'm going to do is start next time with understanding how we're ending here to finish up this part, because I'm going to go to the second one, not the third, next time we meet, I think, but we'll see. Anyway, with that, some version of this, we will see you next time, and I hope you have a good afternoon, morning, evening, and Thanks. Cheers.